Welcome to DNA replication. This is a DNA double helix. To begin the replication process, we will untwist it into a straight ladder. This is the ladder in greater detail. Notice how the strands run in opposite directions and the nitrogenous base pairs are arranged in specific methods. To begin the process, we will insert helicase, our first enzyme. Helicase works by unzipping the DNA into two separate strands. This format is also called a replication fork. Next, we will insert SSBs, the enzyme that keeps each of the strands from recoiling as each strand produces an mRNA. Let's take a closer look at our top strand. First, we will need the addition of a primer, to which our DNA polymerase, the next enzyme in use, can attach itself to. DNA polymerase works in a methodological 5' to 3' direction, adding free nucleotides to its complementary pair as it goes. And voila! Notice especially how the strands are still anti-parallel. This is critical to know because nucleotides can only be added to the 3' end. Now, let's take a closer look at the bottom strand. The same process is repeated, first adding a primer and then adding the complementary base pairs. Remember that because DNA polymerase only works from the 5' to 3' direction, it can be kind of tricky for the bottom strand because it keeps running into the primers. The short chunks it does produce are called Okazaki fragments. As the top strand is one long continuous strand, it is called the leading strand. Because the bottom is made of shorter Okazaki fragments, it is called the lagging strand. At this point, the primers will be removed. And DNA polymerase will repeat the process to fill in the gaps. Though it may not appear so, there are small gaps between where the primer used to be and the mRNA strand. This is where our next enzyme, ligase, comes into play. Think of it as a stapler of sorts. It closes the small gaps between where the primer used to be, or Okazaki fragments, and the rest of the mRNA strand. Finally, the DNA replication process is complete. We know the process to be semi-conservative because as each new double helix forms, one strand will be from the original DNA, and the other strand will be made of the new mRNA. And that's it. Thanks for watching and stay sciencey.